Okay, hello, it's Jubilee again, and we're back for the second part of the final review for this class. Hopefully you guys have already watched the first part of the review. If not, please watch it. I think it's really helpful. Both of these documents that I'm going over with you guys, I have made specifically tailored to your exam. Okay, so I literally look at your exam, I look at the trickiest things that are on it, or the things that I want you guys to understand the most, or that might be tested in more than one question. And I try to make it as straightforward as possible for you guys for both of these documents to really help you with your exam. Okay, so please watch both of these videos entirely, take notes, do whatever you need to do, but please try to learn. Obviously, that's my goal, is to make you learn. But yeah, I had something else to say, but I don't know what. Sorry. I hate that you guys aren't here to ask me questions and whatever. But I will try to cover everything as fully as possible. If you have questions, please let me know. The format of the exam is covered in the previous video, if you'd like to go back and look at that. Hopefully some of the things that we're looking at today in this video will make a lot more sense once you've watched the previous video, okay? And you'll notice every single one of these pictures that I have looks like it's probably a screenshot, and most of them have an A, B, and a C arrow, right? So yeah, they're pretty much screenshots from potential questions you could have, right? And like I said in the last video, I can't guarantee you're gonna have all of these questions. You might have different questions that aren't on this document because of the way the exam is formatted. You just get a random pull of 5 or 6 questions out of a bank of 15, right? So I don't know which ones you're going to get. You might get more of these or less of these, but y'all should all get around the same amount of the ones that I've covered on average. So yeah, hopefully this is helpful for you guys. You guys should recognize a bunch of these pictures when you're taking your exam on Tuesday. So yeah, take some notes, recognize these, and we'll get started. Okay, so this first one is one we're going to go over. You've already seen this picture before, probably, but here you can see, so this is the ovary, right, in the female reproductive system. So this is the ovary, the outer parts, the cortex, inner medulla, right? So here you can see what A is pointing to is this really large looking thing, right, with layers to it, closed up in on itself. It's taking up most of the ovary. And so what this is a corpus luteum. Okay, so A is pointing to the corpus luteum. Remember that the corpus luteum is making estrogen and progesterone. Okay, that's what it makes. It does not make luteinizing hormone. It is created by luteinizing hormone, and that's why it's called the corpus luteum. Okay, B, this is pointing to a secondary follicle in the ovary. Okay, so this would be a secondary follicle, and the reason I know that is because it's not as large as one like this, right? This would be a tertiary follicle or a mature follicle, and it's even labeled like that, a large follicle, but it does have this antrum inside of it, right? So this is an antrum in this secondary follicle, and remember we said the second they have an antrum, they're considered a secondary follicle, and this is going to contain a bunch of the estrogen that the follicle was secreting. Okay. C, what C is pointing to, so this is in the medulla of the ovary, the medulla, the inner part of the ovary, and it's pointing to a bunch of blood vessels. So what it's pointing to would be the medullary blood vessels of the ovary, right? So not the cortical blood vessels, the medullary blood vessels of the ovary. Okay, moving on. So this one is a respiratory system question, and it's basically, it looks like we might be doing some matching here, right? We've got two options of matching, right? And if you look, this one right here is a diagram of what would be pseudostratified columnar epithelium, right? And I know that because there's more than one layer, but they do look column-like, or it looks like it's more than one layer, sorry. That's why it's pseudostratified. And then there's cilia on the top, right? And there's even goblet cells. And so we learned that this is very characteristic. This pseudostratified columnar ciliated epithelium is very characteristic of respiratory epithelium. Okay? And if we look down here, this is actually olfactory epithelium. Okay, this is olfactory epithelium. It doesn't have that same brush border. It is not it's a different kind of epithelium. And even under it, we can see some Bowman's glands and stuff like that, which is characteristic of olfactory epithelium, right? If you look over here, this is respiratory epithelium. So you see how these two look different? We have these really clear cilia up here. And you see how this one looks a lot more like that olfactory epithelium. And these little things sticking up at the top are actually nerve endings of this olfactory epithelium. Okay. So if I were analyzing this, I would say this one does not match this one. This one does not match this one, right? They should be switched. And this one, so these are in the vocal folds, right, where we have stratified squamous epithelium. So this is stratified squamous epithelium, and this is stratified squamous epithelium, right? We've got more than one layers. If you look at the top, those cells are very flat, so that would be squamous. It's stratified squamous epithelium, and so those would match, right? So that's what I would be thinking if I was looking at this picture, trying to imagine what a question about it would be like. Okay, here we are in the testes. So these are in the seminiferous tubules, right? And so that's actually what A is pointing to. So it's pointing to this thing, this big thing, and this big thing, right? Those are those arrows. So these are all the outsides of the seminiferous tubules. So these are seminiferous tubules right here, okay? B is pointing to these bubbly kind of cells in between the seminiferous tubules next to the blood vessels, and those are going to be our Leydig cells. Leydig cells, right? And what those are going to be producing is going to be testosterone, the male sex hormone, okay? So testosterone. These are seminiferous tubules where we're making our germ cells. These are going to be our Leydig cells where we're making testosterone. Let's see, C is pointing to this inside lining of the seminiferous tubules. This inside lining, and this inside lining is what these late spermatids are going to break off of. They're going to break off into the lumen of the seminiferous tubules. And so the reason we call it what it is because of that. So this is called the germinal epithelium. So it's called the germinal epithelium because it's that lining on the inside. It's hard to actually see it in light microscopy. It's just this lining or barrier on the inside of the seminiferous tubules from which the germ cells originate, right? So the late spermatids are going to break off and go into the lumen. So that's our germinal epithelium inside the seminiferous tubules. Okay, this one is talking about the kidneys. And so you can see we have circled here the juxtamedullary nephron and the cortical nephron. Okay, so these are two different kinds of nephrons within the kidneys, right? And you can tell by their name where they're going to be located, right? So cortical nephrons are going to be located more in the cortex, so out here in the kidney. Juxtamedullary nephron literally means next to or beside the medulla. So they're right around here at this barrier between the cortex and the medulla. So juxtamedullary nephrons. Okay, and so they're a little bit different. The main difference is going to be this segment right here, remember, and this is that loop of Henle, this very long loop of Henle in the juxtamedullary nephrons compared to this very tiny loop of Henle in the cortical nephrons. Okay, and so remember we said the reason for the loop of Henle dipping down into the medulla, so that's what it does in the juxtamedullary nephrons, is it dips down into the medulla, and we said the function of that was to create a concentration gradient in the medulla. Okay, and so obviously the cortical nephrons, since they're all in the cortex, do not dip down into the medulla, so they do not help to create that concentration gradient, but the juxtamedullary nephrons do. Okay, and remember we said that concentration gradient is really important for when we get down to these collecting ducts and distal tubule and collecting ducts at the end, because that concentration gradient helps us to concentrate our urine, okay? Because it's the fluid inside the tubules is very dilute, but once we dip it into the medulla where everything's very concentrated, the water wants to leave the tubules and go out into the medulla of the kidney, right? And so what ends up in here is once all the water leaves, we make concentrated urine. Okay, we make concentrated urine. So if I were to ask you which one of these is responsible for helping with the concentration of urine, you would say the juxtamedullary nephron, because they're the ones that make that concentration gradient that makes concentration of that urine possible. Okay, and if I were to ask you which one is more responsible for regulation of blood pressure, okay, so the kidneys help to regulate blood pressure, and the way they do that is by concentrating urine if it's needed, right? So if you're very dehydrated and you haven't been drinking very much, you have less blood in your body, and your body recognizes that, right? You have less fluid filling your blood vessels because you're dehydrated. You don't have enough water filling up your vascular circulation. So your kidney will recognize that. It will say, hey, I'm not getting enough blood and I must be dehydrated. And so in order to try to rectify that, what it does is it will concentrate your urine, save that water, and that helps you to regulate blood pressure. Okay, that's one of the many ways the kidney helps regulate blood pressure. And so again, that's through the juxtamedullary nephron. Okay, and then returning filtrate to the urine. So returning filtrate to the urine, that's where, right, we're forming this filtrate and it's going to end up being excreted as urine. And so both of those would be helpful for that. So both of those do that. Okay. So hopefully that clears that one up. This one is in the liver. So we're in the liver. We're in the center of a classic globule. Okay, so this would be our big this would be our big central vein in the middle of a lobule, right? Our central vein. And so if we look at what A is pointing to the cell lining the inside of this central vein. So just like we learned in the beginning of the semester, this is just going to be an endothelial cell, right? An endothelial cell lining the blood vessel. Okay, so that's what A would be. B is just pointing to this generic cell all throughout the liver, right? And so the main functional cell of the liver is going to be a hepatocyte. So B would be a hepatocyte. Remember, and that's going to have endocrine and exocrine functions, that hepatocyte. C is pointing to this kind of bubbly cell that's sticking out in a little blood vessel, okay? And so C, it's a little hard to distinguish, but I would say it's either a Kupffer cell or an endothelial cell, okay? I would guess Kupffer cell, but you could argue for either, okay? So we're just going to narrow it down to it being a Kupffer cell or an endothelial cell. And remember, Kupffer cells are these specialized macrophages, right? Macrophages of the liver, okay? So they're just eating up junk in there, okay? That was the liver. This is talking about the stomach, right? He even tells you it's the fundus stomach. So A is pointing to these cells right here, these bubbly, almost triangular looking cells. So those would be your parietal cells. Your parietal cells in the stomach. And remember, that's making hydrochloric acid. It's making acid in the stomach, and that acid is necessary to activate all of the enzymes that are being produced in the stomach. okay, for protein digestion. B is pointing to these cells, and remember this is a PAS stain, so they're staining very purple, right? So that means they've got a lot of sugar in them, okay? They're making a lot of sugar. So these would be our mucus neck cells, because remember, mucus is very sugar rich. It's very protein rich. So this is mucus. These are our mucus neck cells, okay? Because they're kind of in the middle, this neck region of the fundus stomach, okay? Mucus neck cells. And then these right here, these are also mucus producing cells on this in the fundus stomach, but they're more on the surface of the fundus stomach. So these would be our surface mucus cells. Surface mucus cells. Okay, they're also very dark staining, right? Because they're very sugar rich from all that mucus. This one is about the skin, right? And hopefully you recognize from my review, this is a picture of a melanocyte, right? So this is a melanocyte. These are not melanocytes. These are cells of the stratum basale, and what they have is a melanin cap. You see how they have all this melanin on the top of them? So that's a melanin cap of the cells of the stratum basale, and that melanin is produced by the melanocytes, okay? And then here you can see this is, again, it even has capping labeled there. So this is just an EM of the melanin capping above the nucleus of the cells of the stratum basale. Okay. This one is in the pancreas. So we're in the pancreas. This is a little blood vessel. Okay, maybe a little venule. You can see the red blood cells in there, so it's a blood vessel. B is going to be a cell of the pancreatic acinar cells. So all of these right here are pancreatic acinar cells, and these little granules that they have within them, these dark blue granules, are going to be those pancreatic enzymes that they secrete, right? Pancreatic enzymes that are really important for digestion, okay? So these are those pancreatic acinar cells. C, so this whole thing right here, these are the cells of the islets of Langerhans, and they're lighter staining. They're not as dark in here, right? So these are those islets of Langerhans cells, and remember, those are producing our insulin and our glucagon. And remember, we talked about the portal system, how they're dropping off insulin and glucagon in the capillary beds right here. It's taken to a secondary capillary bed right here to affect the change, to tell these acinar cells, hey, we either need to start secreting or we need to stop secreting, okay? That portal system. Here we have the eye. So this is just a section of the eye. And you can see here to the left, this is going to be the lens of the eye. So this is that lens right there of the eye. This is going to be the iris of the eye. And this is going to be those zonules coming off of the ciliary body, right? That ciliary muscle. And so remember, the lens of the eye is important for focusing images and directing that light to the photosensitive portion of the eye, the back of the eye, the retina. Okay, so that's what the lens is important for. The iris is kind of like the shutter, remember? So it controls how much light is let into the eye, how much light. So it can open and close to decide how much light is allowed to enter into the globe of the eye. And see these zonules, remember, these are going to attach. It's really hard to cut it perfectly and fix it perfectly, but they're going to attach to the lens, and they're attached to the ciliary muscle down here, and that ciliary muscle is going to contract and relax, and that's going to change the shape of the lens, because remember, the lens is what's going to change shape to focus on an image. Okay, and it happens through these little zonules. Okay, this is the urethra. Okay, and you can see right here, so this is talking about the epithelium lining it, and remember we said in the urethra it has a very special lining, and this is transitional epithelium. Okay, so this is that transitional epithelium that makes it easy for things to expand very easily without causing problems. Okay. Actually, sorry, I think this is a ureter, not a urethra. I don't know what I was thinking, but either way, still transitional epithelium, ureter. This right here is talking about the connective tissue under that transitional epithelium. And so remember, that's the lamina propria, that's the name of it. Connective tissue under the epithelium in most organs, lamina propria, is that connective tissue. And then out here, all of this really thick pink stuff, this is going to be smooth muscle. Smooth muscle, not skeletal muscle, right? This is all smooth muscle out here. C. Okay. This one, so we're back in the stomach. So A is pointing to those mucous neck cells, right? So if I were to ask you what they produce, hopefully you would tell me they make mucus, right? These are those mucous neck cells. So these are producing a bunch of mucus. Here are those little mucin granules, okay? B, this is a parietal cell. These are parietal cells, and remember we said they make hydrochloric acid. They also make bicarbonate, but the main product of them is going to be hydrochloric acid. Okay. C, this is a fibroblast. So this is just a little fibroblast making connective tissue, right? And so if we try to remember what they excrete or what they make, that would just be collagen, right? They're constantly making collagen, connective tissue. So that's a fibroblast. Okay, here we are in the testes again of male repro. So this is inside a seminiferous tubule, this is outside. All right, and so A, what we're pointing to, so we're in the seminiferous tubule, but we're a little ways inward. So this would be either a primary or secondary spermatocyte. Okay, and you can tell because it has these little meiotic figures going on in here. Right, it's undergoing meiosis, and then it's also a little further inward, right? It's past the barrier that this cell makes, and hopefully that gives you a hint for what this cell is. But this is a Sertoli cell, okay? And you can see that this is a Sertoli cell because it's branching and it's irregular. It's branching around to the cells next to it, and it's at this level in the seminiferous tubule, kind of like one cell off the base. The Sertoli cell. And our spermatocytes are always going to be within the Sertoli cell, so more inwards toward the tubule than the Sertoli cell. Down here would be a spermatogonia, right? There's no arrow pointing to it, but just so you know, that's a spermatogonia. They undergo mitosis. This is that lining of the seminiferous tubule with some myoid cells. And then out here, outside of the seminiferous tubule, these would be Leydig cells. Leydig cells, and remember those are the ones that are producing testosterone, the male sex hormone. Okay, so these would be Leydig cells, and you can even see their little secretory granules of stuff they're making, the testosterone they're making. Okay, this is a digestive system question. Okay, this is a digestive system question, sorry. And so what we basically have is 3 different pictures of 3 different organs. Okay, so if you try and identify what organs these would be, so this is an EM, so it might be a little hard to tell, but he even gives you that it has microvilli, right? So these are the microvilli of a cell, and up here would be a goblet cell. Okay, so what cell has a bunch of microvilli? That would be the small intestine. Right, so these are the cells of the small intestine with all their microvilli. And if we remember the function of the small intestine in the digestive system, its main role is going to be absorption of nutrients. Right, so this is the small intestine. Its main role is absorption of nutrients. B, so this one right here, so we've got stratified squamous epithelium up here, a bunch of skeletal muscle, a bunch of smooth muscle. So this tubular organ is going to be our esophagus, right? Because we've got stratified squamous epithelium. We expect there to be a lot of friction in the esophagus, right? And that's because the main function of the esophagus is movement of food, right? Just movement of food from one place to the other. So this is the esophagus, and that's its function in the digestive system. This one right here, So this one right here, these are cells of the, sorry, I'm being distracted. Okay, these are cells of the stomach. Right? These are our chief and parietal cells. And remember, we can see all of these little granules in here. And remember, in the parietal cells, those granules are from the mitochondria. And that's because it takes a lot of energy to produce hydrochloric acid. And in these chief cells, the granules are from the pepsinogen that they're making. Okay, and remember, one of the main functions of the stomach is secretion of enzymes, right? So we're secreting that pepsinogen, which is an unactivated enzyme, and then it gets activated by the hydrochloric acid. Okay, so secretion of digestive enzymes. This one, so this is the gallbladder. This is the gallbladder, and so you can see out here on the outside of the gallbladder, so this is going to be a bunch of smooth muscle. Okay, out here of the gallbladder. B, this is going to be that connective tissue, that lamina propria of the gallbladder under that epithelial lining. And C, this is going to be that epithelial lining of the gallbladder, so that simple squamous epithelium. This one is the retina. So these are the layers of the retina, right? So we're just identifying them. So A, we can see, so this is that outermost layer of the retina, and that's going to be the ganglion layer, right? The layer that eventually turns into the optic nerve. So this is the ganglion layer. This next layer of nuclei is going to be the bipolar cell layer, and remember, they're transmitting the signal from the rods and cones to the ganglion layer. So bipolar cell layer. This would be the photosensitive layer, the rods and cones, pigmented epithelium, right? But those aren't labeled. So this one right here is pointing to the layer under the retina with all the blood vessels, right? So this is the vascular layer. So that's the choroid layer. Remember, the choroid layer with all the blood vessels and everything. Okay, so this is the pituitary. And so it's just pointing out the three different areas of the pituitary, right? So this is all pars distalis with our acetophils and basophils. This is our pars intermedia with all our blood vessels and whatever. This is our pars nervosa, right, with our herring bodies and pituocytes and whatever, right? So just remember what hormones are being secreted, right, by these acetophils and basophils, what hormones are stored in these herring bodies, and nothing, as far as y'all know, is secreted in the pars intermedia. Okay, this one is talking about secretion of the cell. So what trips up a lot of people is that this cell is actually showing two different methods of secretion, right? So a lot of people see this one, this is really obvious, right? So we're losing a part of the cell membrane every time one of these little lipid droplets buds off, right? We're losing a part of the cell. So that would be apocrine secretion, okay? But this one is actually undergoing exocytosis, right? So we're not losing any of the cell when we do this. It's just normal exocytosis. So that would be merocrine secretion. So this cell is actually showing apocrine and merocrine secretion, right? And then this is just an EM of a cell actually doing it, okay? This is the diagrammatic view. This is the same thing, just an EM of it actually happening in a real cell. Okay? So this is the fundic stomach. And so A, what it's pointing to are these kind of more regular shaped cells. Those would be the chief cells in the fundic stomach. So these are all chief cells. And the way that I know that is because they're not these cells, right? And these are the parietal cells that are very triangular looking. They look like soft triangles to me. Okay, so that's a very characteristic parietal cell. So I know that these are chief cells. Okay, and remember, chief cells are making your pepsinogen, parietal cells are making your hydrochloric acid, and then every once in a while you have a cell like this one that C is pointing to. So you see how the granules are way different color in this cell, and the granules are a way different color in this cell, even in this stain. Every once in a while you have these cells. These are just special cells in there, and those are called enteroendocrine cells. Enteroendocrine, okay? And those make histamines. Okay, those enteroendocrine cells. Okay, here we are in the liver. So this is the liver. So this is a big hepatocyte with a bunch of things going on in it, right? A bunch of secretions and stuff. And so this is the little sinusoid. So this is where the blood's going through. Sinusoid of those sinusoidal capillaries, right? So the blood is going all through here. So this is that vascular space around the hepatocyte, okay? B is talking about this kind of space right here of interdigitation. And this is, remember, that space of Dease. So it allows for the blood to go through that sinusoidal endothelium, right, with big gaping holes in it, and enter the space of Dease so it can bathe the hepatocyte in blood, right? So that's that space of Dease. And right here, he blibbed out the little label, but this little kind of star-shaped thing in between two cell membranes of hepatocytes, this is going to be a little bile canaliculi, right? Bile canaliculi. And remember, that's because hepatocytes have endocrine and exocrine functions, and we don't want them to mix, right? So all our endocrine secretions would go out here into the blood, and all of our exocrine secretions would go into this little bile duct, this little bile canaliculi, eventually to a bile duct, okay? Proximal convoluted tubules. So all of these are examples of proximal convoluted tubules. Remember, we said that in the other review. And remember how we know that is it has this big brush border right here, and these irregularly spaced out nuclei. Okay, so just know all of these are different examples of proximal tubules with their brush border. This one, so this is in the respiratory system, and this is an example of our olfactory epithelium. So remember, this is olfactory epithelium. It's a special type of epithelium, and we've got our little nerve projections, nerve endings coming out the tops of it. Okay, B is pointing to these darker areas, these darker little globules, and remember, those are our Bowman's glands that are making the mucus that goes up here on our olfactory epithelium. And then C, these kind of lighter staining bundles. Remember, these are our nerves. These are our little nerve bundles, right? Because we've got nerves, nerve endings up here trying to sense smell, right? Smell things. Okay, this is male repro. So here we're looking at, it even tells you that's transitional epithelium. Oh, sorry, that's not what the arrow is pointing to. A. So A is pointing to this kind of dense area around the erectile tissue, right? So we've got two different types of erectile tissue in the male right here. And we've got this dense, irregular connective tissue around that erectile tissue. And remember, that's just to help give shape to the penis so that it maintains that rigid tubular shape as opposed to blowing up like a balloon. So that's why we need all that connective tissue there, that dense irregular connective tissue. B, these are pointing to little nerve bundles. So these are little nerve bundles for feeling and whatever right there. And then C, what this is pointing to, so this is going to be smooth muscle all around the outside of the penis. Okay, so that's just a bunch of little smooth muscle fibers up there. This one, so this is female reproductive system, and so this is all the organs of the female reproductive system basically, right? So we've got the ovaries, it's caught by the infundibulum, and then it goes through this structure. It's the fallopian tube or the uterine tube. Okay, fallopian tube right there, so that's A. B, so that's the uterus, right? That's the womb. That's where the fetus is going to implant, right? So that's the womb, the uterus. C, it's pointing to this barrier between the uterus and the vagina. This down here is the vagina, right? Uterus, vagina. So this barrier in between them is going to be the cervix, okay? So that's the cervix. So this one right here, this is the retina, right? And you can see here in this part of the retina, this is a special area of the retina where the top two layers, the ganglion and bipolar cell layer, are pushed aside. They're pushed aside, so you see how it gets flat, and then we've just got these rods and cones, right? The photosensitive layer right here. So this is that special portion of the retina. It's called the macula. The macula. And remember, the point of that is so that you can have the sharpest vision at this point, right? So we're pushing these layers out of the way so the light can directly hit down on these rods and cones. So it can directly hit those rods and cones. Okay, and we also have more cones in this area as well because this is where we see the most detail. So this is the macula of the retina, and this is normal retina. Okay, this one is also the eye. So A in the eye, so it's this innermost layer, the back of the eye, so that's going to be the retina. So this is the retina of the eye. B, it's pointing to this layer with a bunch of connective tissue deep to the retina and the choroid layer. So that's going to be the sclera, right? The sclera with a bunch of dense, irregular connective tissue, and that's part of the supporting layer, right? The supporting layer of the eye. A is obviously part of the photosensitive layer. C is pointing to this big space in the middle of the eye, and remember that is the posterior chamber of the eye, and it contains vitreous humor. Okay, so vitreous humor, posterior chamber of the eye for C. Okay, this one, remember that exact picture from the other review, right? This exact picture with this white circle. So remember we said this is in the thyroid gland, and this is a parafollicular cell, right? These would all be follicular cells, and in between the follicular cells would be parafollicular cells. And remember what we said the parafollicular cells make is going to be calcitonin. And remember we said the function of calcitonin is to tone down the calcium levels. And so how it does that is by inhibiting osteoclasts, right? It inhibits osteoclasts from breaking down bone, because when they break down bone, they put more calcium into the blood, right? So if we want to tone down calcium, we'll inhibit those osteoclasts. And so if we look at this picture, so these all go with the same question. So if we look at this picture, this is a depiction of osteoblasts. So these are osteoblasts. They're by themselves. They're lined up on that osteoid, ready to build more osteoid. They're building, they're laying down bone. This is a picture of an osteoclast. And you can tell because it's very clearly multinucleate and huge, starting to break down bone and osteoid. And then these are little osteocytes, right, in their little lacunae in the osteoid. So those are osteocytes, right? So if I were to say, what cell do these parafollicular cells affect, you would say the osteoclasts, right? And the osteoclasts would be B. Okay. So here we go. This is one of the same pictures I showed y'all in the other review. This is these are the glands in the skin, right? And so A, this is pointing to a gland that is emptying into the hair follicle. It's got these really big bubbly cells. Those are going to be your sebaceous glands. And remember, those are going to do holocrine secretion. The whole cell is the product, holocrine secretion. These are sebaceous glands. B and C are pointing to the same type of gland. So they're the same type of gland, and so these are both eccrine sweat glands, your normal sweat glands. Remember, so these are eccrine sweat glands, and they do merocrine secretion, okay? This one is the large intestine, and so this is the outside of the large intestine, and we can see we've got these two different layers of smooth muscle. These are two layers of smooth muscle, so one's going this way and one's coming at you. So A would be pointing to those two layers of smooth muscle, and remember we call that the Muscularis Externa. Muscularis Externa. And remember, we said the nervous tissue, or the nerves in between these two layers, is Arbox plexus. Arbox plexus, and that's what's controlling these two layers for peristalsis of the large intestine. And then the single cell layer on the very outside of the large intestine, that's going to be our mesothelium, right? Our mesothelium, that type of epithelium that lines all organs. Okay, here's another one. It looks like we're doing some matching, right? So A, that is a picture of the salivary glands, and here it is pointing to the salivary glands. B, this is a picture of the liver, right? There's our classic lobule with our central vein. So this is a picture of liver tissue, and what it's pointing to is the gallbladder, right? So those are different things. These were the same things. This is a depiction of the pancreas. We've got all these pancreatic acinar cells and these islets of Langerhans. And what it's pointing to right here is the large intestine, right? Not the pancreas. So those also didn't match. Okay. Here, so this is a glomerulus in the kidneys. And so A, right here, is going to be pointing to the vascular pole of the glomerulus. So this is where all the blood is entering the glomerulus and leaving the glomerulus after it gets filtrated. Right, so this would be the vascular pole where all the blood's entering. And then it gets filtered out of this glomerulus and it leaves through the urinary pole. Okay, so blood enters and leaves here, but some of it gets put into the filtrate, and so that enters the urinary pole, right, and goes into the proximal tubule and everything like that, and then is filtrate. Okay, and then C right here, so this is pointing to like the outside cells of the glomerulus, the outside lining cells, and those, remember, are going to be our parietal cells. Our parietal cells lining the outside of here, okay? And then the cells in here would be the podocytes. These would all be the podocytes, okay? So this is in the lung, and A is pointing to this very bubbly cell, this very big bubbly cell in the parenchyma of the lung. So this is going to be a type 2 pneumocyte, remember, because it's very fat, it's bubbly, and that's what's going to be producing our surfactant. So this would be a good example of a type 2 pneumocyte. B, so these are these big cells with a bunch of granules in it projecting into the alveolar space. So that's going to be an air macrophage or alveolar macrophage. So these are little macrophages in the lungs, just picking up dirt and debris and everything. And then C, this is pointing to a very flat cell in the lung. And remember, we have two main types of flat cells that we worry about in the lung besides our normal fibroblasts. And those are going to be type 1 pneumocytes, and then we also have endothelial cells, right? But you can tell this is an endothelial cell and not a type 1 pneumocyte because you see what it's lining. It's lining a blood vessel, right? This is a blood vessel with little red blood cells in it. So this is an endothelial cell, not a type 1 pneumocyte. Okay, this is male, this is a diagram. And so A, this is pointing to the seminal vesicles. The seminal vesicles that are going to help to add fluid and nutrients and whatever to the ejaculate. B is pointing to the ejaculatory duct. The ejaculatory duct that exits and enters the penis. The C is going to be pointing to the prostate gland, so the large prostate gland that is also going to help add fluids, nutrients, buffers, whatever to the ejaculate. This is female, so we're talking about the follicles in the female ovaries. And so A, this one right here, we've got a big oocyte with some flat follicular cells around it, right? They're very flat, they haven't started dividing yet. So that's going to be a primordial follicle, And remember that primordial follicles are present at birth, okay? B is going to be pointing to a primary follicle. We haven't cut it through the center where we can see the oocyte, but you can see the follicular cells on the outside are very bubbly and expanded and they're starting to grow. That would be a primary follicle. see right here, so we can see now we have an antrum. We're starting to form an antrum. So the second we have an antrum, this is a secondary follicle. Okay, secondary follicle. Here we've got more of an ovary, so this is a tertiary follicle, a huge mature tertiary follicle. And we can see A is pointing to that oocyte, that ovum in the follicle. These would all be granulosa cells, right? And then B is pointing to that layer under the granulosa cells, and that would be our theca interna. Theca interna. And then remember, outside of the theca interna is the theca externa. So it's just these two different layers outside of the granulosa cells. Okay. This one is an EM of a spermatozoa. And so A is pointing to this outer layer. So this is going to be the cell membrane of that sperm cell, that spermatozoa. So that's the cell membrane. B right here, so this is the acrosome right here. So B would be pointing to the outer acrosomal layer. The outer acrosomal layer. This one would be the inner acrosomal layer. Inner acrosomal layer. And then right here, this is pointing to those microtubules in the tail. These are the microtubules within the tail. And right here, they are singlets. Okay, eventually they turn into microtubule doublets, but right here it's a little singlet in the cross section of the tail. Okay, and so that's all the images that I have for you guys. Hopefully this was helpful. Thank you guys so much for watching. I appreciate it. And honestly, I just wish you guys the best of luck on your final, on all your other finals for the rest of the semester, for the rest of your school career. All I want y'all to do is learn. All I want y'all to do is succeed. So good luck with that. And I hope you guys enjoy your summer. It was my pleasure teaching y'all.